Everyone, and good afternoon everyone, and welcome to the NEET biology lecture series. So today is the day 3 for the biology lecture NEET series, and this is a 60-day crash course. All right. So today we'll be studying the chapter 2 of the NCRT book, which is the biological classification. Okay. And this is the part 2 of the lecture. In the previous lecture, we have already studied about the various system of classification. Okay, we were studying about the kingdom Monera and kingdom Protista, and today we will cover these topics. So first topic is the basic introduction of fungi. Okay, so we will study about the general features of the fungi, and then we will study about the different phylum of the fungi. Okay, we will study about the Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes in detail. Then we will study about the next kingdom, which is the kingdom Plantae, and then we will study about the kingdom Animalia, and then we will study about viruses, viroids, prions, and lichens. And then we will be solving some of the previous year NEET questions. Okay. Now again, I'll give you the weightage of this chapter. So from this chapter, biological classification, around 1 to 2 questions come in the NEET exam. But as I've already told you earlier, this is a very important topic. You need to study this topic in detail. Every topic is important from this chapter. Now let's start with the introduction of the kingdom fungi. Okay, so I would like to mention that all the topics from this in this lecture are from the NCRT book. Alright, so this is completely NCRT based lecture, and we are studying the 5 kingdom classification in detail. Okay, so 5 kingdom classification. What were the kingdom? If you remember, I'll repeat it. First was the kingdom Monera, second was the kingdom Protista that we have already covered in the previous lecture. Today we will be studying kingdom fungi, Plantae, and Animalia. Okay, so let's start with the brief introduction of the kingdom fungi. Now the fungi constitute a unique kingdom of heterotrophic organism. Alright, you need to remember that they are heterotrophs, means they feed on or depend on other organism for food. Okay, now there is a great diversity in morphology and the habitat of fungi. Okay, they are very diverse, and the habitat also is very diverse, means the environment which they live in. Alright, now the common mushroom that you eat. Okay, we eat mushroom daily in our life. Okay, the mushrooms and the toadstools, they are also fungi. Okay, so they are edible fungi. Now some of the unicellular fungi, so these are the some of the benefits of the fungi that I have listed. Okay, some unicellular fungi such as yeast. Alright, so most of the fungi are multicellular, but there is one exception, yeast. Scientific name for yeast is Saccharomyces cerevisiae. Okay, so this yeast is unicellular eukaryote. Okay, they are used to make bread and beer. Okay, so these are the benefits of fungi. Now another benefit, another advantage, another importance of fungi is that some fungi are source of antibiotics. Okay, example is penicillium. So penicillium is a fungus, okay, from which penicillin antibiotic is obtained. Okay, so penicillin. So this antibiotic is obtained from penicillium. Okay, there are different species of penicillium. Okay, one is Penicillium notatum. From this also penicillin is isolated. Another one is Penicillium chrysogenum. Okay, from this also penicillin antibiotic has been isolated. Now here I have listed some of the disadvantages or the harmful effect of the fungi. So some of the fungi are parasitic also. They cause damage to the host organism, okay, or they feed on the host organism, and and this is their survival strategy. Now you must have seen white spots on the mustard leaves. So this white spot is caused due to a parasitic fungus known as albugo. Okay, so albugo is a example of a parasitic fungus. Albugo candida is the full scientific name. Okay. Now other fungi also cause diseases in plants and animals. Okay. Example is wheat rust causing Puccinia. Okay. So Puccinia is the fungus here. So Puccinia causes wheat rust disease. And this infection by the fungus leads to lots of yield loss. Okay. That's why we need to control the infection by this fungus. Now, what is the habitat of fungi? The fungi can survive in, or they can live in air, water, soil, and they can even live on animals and plants. Okay, animals and plants can also be the host of the fungi. Now let's study about the characteristic of fungi, some general characteristic of fungi. Now, as I've already told you, yeast is an exception. It is unicellular. Okay, most of the fungi are filamentous and multicellular. Okay, they are filamentous, means they have hyphae or mycelia. Now the body of the fungus, okay, so this is an image of a mushroom. Okay, so mushroom is also a fungus. So the bodies of the fungi consist of long, slender, thread-like structures called hyphae. Okay, so this you have to remember. Fungi consist of thread-like structure, or they are made up of thread-like structure known as hyphae, and the network of hyphae, okay, is known as mycelium. All right. So here in this figure you can see mycelium here, which is a network of hyphae. These are the hyphae individual one. Individual ones are hyphae, which are thread-like structure. All right, and network of hyphae is mycelium, and this is the fruiting body. Okay, fruiting body consists of spores. Okay, so the fruiting body consists of spores, or you can say they bear spores, which help in the asexual reproduction. Now the hyphae can be of two types. First is septate hyphae, and second one can be cenocytic hyphae. Now what is septate hyphae? Okay, this is an image showing the septate hyphae. So hyphae, so septate hyphae have septate or cross wall in their hyphae. So here you can see the septa. Okay, these are the cross walls present in the hyphae. So these one are known as septate hyphae. Okay, you can see nucleus here. Okay, and then cytoplasm is present. All right, so this is known as septate hyphae. Second one is cenocytic hyphae. Cenocytic hyphae is also known as aseptate hyphae, and there is no septa present. So this septa is not present. You can see here no septa or cross wall is present, and because of the absence of the septa, it becomes multinucleated. Okay, here you can see there are a lot of nuclei. One nuclei, second nuclei, third nuclei. Okay, like this nuclei are present. Now again, coming back to the characteristic of fungi, let's talk about the cell wall of fungi. So they are unique. Cell wall of fungi are unique. They are composed of chitin and polysaccharides. Now let's talk about the nutrition of fungi. So fungi they can be heterotroph, means heterotrophic, depend on other organism for food. They can be saprophytic, means absorb soluble organic matter from the dead substrate, or you can say they simply feed on dead and decaying organism. Now they can also be parasitic, means they depend on living plants and animals, okay, for food. Alright, they are dependent on another organism for food. Next is symbiotic. They also show symbiosis. Okay, what is symbiosis? Symbiosis is mutual interaction, okay, also known as mutualism. Okay, so it's a beneficial interaction. Both the organisms are benefited from each other. Okay, so they are in symbiotic relationship. So example is this. So fungi can be in symbiotic association with the algae. Okay, so when algae and fungi come together, they form lichens. Okay, so algae here is a phycobiont. Algae is known as a phycobiont. Whenever we use this term phyco, it refers to algae. Okay, and fungi. Fungi is a mycobiont. If we talk about fungi, it is a mycobiont. Myco means fungi and biont. Okay, now fungi can also be associated with the higher plant, the roots of the higher plant. Okay, and this association or interaction is known as mycorrhiza. Myco means fungi, rhiza means roots. Okay, rhiza means roots. Now let's talk about the reproduction in fungi. So fungi can reproduce by vegetative reproduction, which is also a sort of asexual reproduction. Okay, they can reproduce by asexual reproduction, and they can also reproduce by sexual reproduction. Okay, so first one is vegetative reproduction. Okay, so they can either reproduce by fragmentation, fission, or budding. All these three come under vegetative reproduction. Okay, so what happens during vegetative reproduction? Okay, the offspring forms are genetically similar to the parent. Okay, and only one parent is involved. Okay, in both vegetative reproduction and asexual reproduction, one parent is involved. This you have to remember. Only one parent. That's why it is known as asexual reproduction, whereas in sexual reproduction, two parents are involved. Okay, now let's talk about fragmentation. Fragmentation, what happens? The plant part, okay, such as root, leaves, or stem, okay, they develop into an entire organism. This is known as fragmentation. Fission, you already know, cell breaks or divides into two. Budding, okay, a bud formation takes place, and this leads to the formation of a new organism. Now let's talk about asexual reproduction. It takes place with the help of spores, okay, which are haploid. So spores called conidia, okay, which are haploid N, or sporangiospores, or by zoospores. Okay, so these all are asexual spores. Okay, now let's talk about the sexual reproduction. Sexual reproduction can take place with the help of oospores, ascospores, and basidiospores. So these are sexual spores which help in sexual reproduction. Okay, now the various spores, okay, are produced in distinct structure called fruiting bodies. All right, so fruiting body. So this is the fruiting body. So this is an image of a mushroom. You can see mycelia here. All right, this is the stalk. Now fruiting body. This is entire is the fruiting body. This is the cap, and here the spores are present. Okay, so spores are present here. This is a spore forming part, also known as sporophore. So fruiting body bears the spores. Now let's study about the life cycle of the fungi. So this is the general life cycle of the fungi. Now the sexual cycle involves the following steps. If we talk about the sexual reproduction, we are talking about the sexual reproduction here. So first is the plasmogamy, second is the karyogamy, and then we have the meiosis, zygotic meiosis, okay, which reduce the number to half. All right. Now let's study about this plasmogamy. So these are the stages of the sexual reproduction. First is the plasmogamy, which means the fusion of the protoplasm between two motile or non-motile gametes. All right. And when we talk about gametes, we are talking about sexual reproduction. If we talk about spores, we are talking about asexual reproduction. Now second is karyogamy, which is the fusion of the nuclei. Okay, in second stage, there is fusion of nuclei. In first stage, there is fusion of protoplasm. And in third stage, what happens? Meiosis takes place. Okay, also known as zygotic meiosis. Okay, and this results in the formation of haploid spores. Okay, now again let's study about the reproduction in fungi. So what happens when the fungi reproduce sexually? So what happens? Two haploid hyphae of compatible mating types come together and fuse. So what happens? Two haploid hyphae, this you have to remember. What fuses together? Two hyphae fuses together. All right, one is known as plus and one is known as negative. So both these hyphae, they fuse together. All right, this is also haploid, this is also haploid, and what happens here? A dikaryon is formed. Okay, so both these, okay, a dikaryon is formed, a cell which consists of two haploid nuclei. Okay, this is known as dikaryon. So in some fungi, the fusion of the two haploid cell, okay, these two cell, fusion of the two haploid cell immediately results in a diploid cell. Okay, so sometimes this dikaryon stage is not formed. Instead of that, a diploid cell is formed. Okay, immediately after fusion, which will be 2n, obviously. Okay, diploid cell are 2. Okay, they consist of two sets of chromosome. So after this, usually this dikaryon is formed. Okay, this is known as dikaryon. Okay, usually this stage is formed, but sometimes this stage is skipped and a diploid cell is formed directly from here to here. Okay. Now, however, in other fungi such as Ascomycetes and Basidiomycetes, an intervening dikaryon stage, this I have already told you, which is n plus n, that is 2 nuclei per cell, occurs, and such a condition is called a dikaryon, and the phase is called dikaryophase. Okay, now after the dikaryon phase of the fungi, the parental nuclei fuse and the cells become diploid. Okay, so this was dikaryon consisting of two nuclei, haploid nuclei, and they fuse together. Okay, and this is known as karyogamy. Fusion of the nuclei takes place and a diploid cell is formed. Okay, how this is formed? By karyogamy. Okay, this is fusion of nuclei and a diploid cell is formed. And what will happen? Third stage was meiosis. Okay, meiosis will take place now. The fungi then forms the fruiting bodies in which the reductional division occurs. Okay, now reduction division occurs where? In the fruiting bodies. We will discuss these fruiting bodies when we will study Ascomycetes and Basidiomycetes. Both have fruiting bodies. Okay, now the reduction division takes place in the fruiting bodies. Meiosis takes place in the fruiting bodies. Meiosis was the third stage. Now, and this leads to the formation of haploid spores. Okay, this leads to the formation of haploid spores. Now, the fungi has been classified further. Okay, and on what basis it has been classified further? First one is the morphology of the mycelium, whether it is septate or aseptate. Second is the mode of spore formation, means whether the spores are formed exogenously or endogenously. And third is the fruiting bodies. Okay, now let's study about the different phylums within the kingdom fungi. First one is Phycomycetes, second one is the Ascomycetes, third is the Basidiomycetes, and fourth is the Deuteromycetes. We will study all these phylum in detail. Now, what are the general characteristics of Phycomycetes? Let's start with Phycomycetes. Okay, so what is the habitat? Now, they survive in or they live in aquatic habitats. They can live on decaying wood, okay, in moist and damp conditions, and they can also live as an obligate parasite. Now, what is obligate parasite? Okay, so these organism which is known as obligate parasite, they need a host to complete their life cycle. Okay, they cannot survive outside the host. They need a host organism to complete their life cycle. Okay, now second is mycelium can be aseptate or cenocytic in members of Phycomycetes. If we talk about the reproduction, all right, asexual reproduction, Phycomycetes can reproduce by asexual reproduction. Okay, so asexual reproduction takes place by, I've already told you, by zoospores which are motile, or by planospores which are non-motile. Okay, so question can come from the exam. Okay, in Phycomycetes, how asexual reproduction takes place with the help of which kind of spores? Okay, or which kind of asexual spores? Then you have to say from zoospores and aplanospores. Now, these spores are endogenously produced. Okay, I've told you what were the bases of the classification. First one was the type of spore formation. They are formed endogenously in the sporangium. Now what is sporangium? Sporangium is a structure in which spores are produced. Okay, this is known as sporangium. Now they can also reproduce by asexual reproduction. Okay, and during sexual reproduction, what happens? A zygospore is formed. A zygospore is formed by the fusion of two gametes. Okay, if we are referring to gametes, we are talking about sexual reproduction. Now these gametes can be similar in morphology. Okay, if they are similar in morphology, these two gametes, then it is known as isogamy, isogamous. And if they are dissimilar in morphology, means the size is different. Okay, then it is of two types, anisogamous and oogamous reproduction. Okay. Now some of the example of phycomycetes. First one is the mucor. So mucor under microscope looks like this. Okay, you can see the mycelia here. This is the sporangium. Okay, this is the sporangium. Again here rhizopus is the second example. Rhizopus is also commonly known as bread mold. Then we have albugo, which cause the white spots on the mustard leaves. Okay, albugo, the parasitic fungi on mustard, it causes white spots on the mustard leaves. Now let's talk about the ascomycetes. So ascomycetes is also known as the sac fungi. Okay, why they are known as sac fungi? Because the asci, the ascus which are formed, okay, is sac shaped here. Okay, I will tell you what is ascus in upcoming slides. For now, you have to remember that they are known as sac fungi because the ascus. And what is ascus? It is a spore bearing structure. Okay, spores are produced in the ascus, spore bearing structure. Okay, and this ascus is sac shaped. That's why ascomycetes are known as sac fungi. Now let's talk about the general characteristic of ascomycetes. So they are multicellular. Example is penicillium, and they are rarely unicellular. Example is Saccharomyces. Saccharomyces is what? Saccharomyces is yeast. Okay, so this is an exception in kingdom fungi. Now what are the general characteristics of ascomycetes? Nutrition. Okay, it can be saprophytic. They can be decomposers. Nutrition can be through parasitic mode, or they can attain nutrition by growing on dung also. Okay, when they grow on dung, then they are known as coprophilous or coprophilous. Okay. Now let's talk about the reproduction in ascomycetes. So they can reproduce first by asexual reproduction. Now the asexual spores are conidia here. Okay, if someone asks you in the exam, if there is a question, what are the asexual spores produced in ascomycetes? Conidia, which are produced exogenously on a special mycelium called conidiophores. Okay, so here is a conidia which is born on a conidiophore. Okay, this is a conidia which is produced exogenously on conidiophore. So this is the conidiophore. Okay, now the conidia on germination produce mycelia. Okay, now reproduction, ascomycetes can also reproduce by sexual reproduction. Now the sexual spores are called ascospores. This you have to remember. In ascomycetes, sexual spores are known as ascospores, which are produced endogenously. Okay, which are produced endogenously in a sac-like asci or ascus. Okay, sac-like. That's why they are known as sac fungi, ascomycetes. Now this is the general life cycle of ascomycetes. This we have already discussed. Okay, this is a sexual phase. This one is the asexual phase. Okay, so what happens? Here you can say this is the dikaryon. Okay, dikaryon, and this fuses to form a diploid cell. So let's start with here. Hypha. Two hypha, they fuse with each other. Okay, two hypha of plus strain and minus strain. They fuse together with each other, leading to the formation of dikaryon. So this is a dikaryon, N plus N. Okay. Then what happens? There is a nuclear fusion. Nuclear fusion leads to the formation of diploid cell. Okay. And then meiosis takes place, leading to the formation of haploid spore. Okay. So this cycle keeps on going on. Okay. So this haploid spore will then germinate to form hypha. Okay. And then again two hypha of plus and minus strain. Okay. They will fuse together first. So here plasmogamy will take place. First plasmogamy. I'm writing P for plasmogamy. Then we have karyogamy, nuclear fusion. This is karyogamy, K. And then we have the meiosis, M. So you can remember P, K, and M. Okay, if we talk about asexual reproduction, then okay, yeast is an example, which is unicellular. So what happens? They produce conidia, okay, or mitospores. Conidia produced exogenously, okay, and these conidia germinate to form the hyphae or mycelia, you can say. So asexual cycle is very simple, okay. So the hypha, okay, forms the conidia on the conidiophore, okay, and then it germinates to form again the hypha or the mycelia. Now some of the example of ascomycetes, aspergillus is a very common example of ascomycetes. This is very important, aspergillus. Then we have claviceps, and then last one is neurospora. Okay, so this is the image showing aspergillus. Okay, these are the spores. All these are the spores. Okay, and these spores are born on the conidiophore. This is the conidiophore. Okay, then we have claviceps, which looks like this, and this is the neurospora. Okay, what is the importance of neurospora? So neurospora is, first you have to remember, it is an haploid organism, N. It is an haploid organism and it has lot of importance in the genetics. Why? Because neurospora is very easy to grow. It is very easy to isolate. And what happens? Being haploid, we can express lot of mutations here. Okay? Because the dominant gene is not present here. The complementary gene, another gene is not present because it is haploid. Okay? So we can express lot of mutants or lot of mutations here. Okay, mutations are generally recessive, you have to remember. So being haploid, lot of mutation can be expressed here. So that's why it is used in genetics studies. Now let's study about the economic importance of ascomycetes. Okay, so neurospora is used extensively in biochemical and genetic work. This I've already explained. Many members like morels and truffles are edible. Okay, they are edible and considered delicacies. Now let's talk about the basidiomycetes. Now the commonly known form of basidiomycetes are, this you have to remember, mushrooms, bracket fungi and puffballs. All these are included under basidiomycetes. Now let's study about the harmful effect of basidiomycetes. Okay, so they usually, what is the habitat of basidiomycetes? They grow in soil, all on logs, all on tree stumps, and in living plant bodies as parasites. So this is the harmful effect of basidiomycetes, and they cause rust and smut diseases in plants. Now what are the general characteristics of basidiomycetes? Mycelia can be branched and septate. Asexual spores are generally not found in basidiomycetes. This you have to remember. But the vegetative reproduction takes place by fragmentation. Okay, vegetative reproduction is common, which is also a type of asexual reproduction. Okay, so here if someone asks you, you have to say vegetative reproduction or fragmentation takes place in basidiomycetes. And the spores are absent. Okay, asexual spores like conidia and all are absent. Now another unique feature of basidiomycetes is that the sex organs are absent here. Okay, but plasmogamy, fusion of the protoplasm, is brought about by fusion of two vegetative or somatic cells. So two vegetative or somatic cells, okay, of the hypha or the mycelium, okay, they fuse together Okay, again plus and minus strain. Here they are saying of different strains, plus and minus cells, or you can say somatic cells, or the hyphae, they fuse together, which is known as plasmogamy. Now again the resulting structure is a dikaryon, okay, consisting of two nuclei, n plus n. Okay, so the two strains, okay, which are haploid, they fuse together to form a dikaryon, n plus n. This is a dikaryon. Okay, so the resultant structure is dikaryon or dikaryotic, which ultimately give rise to basidium. So basidium is a structure bearing spores. Like in ascomycetes we had ascus, in basidiomycetes we have basidium, which is club shaped. Okay, so this basidium is club shaped. Okay, that's why basidiomycetes is also known as club fungi. Now if we talk about the reproduction, first one was the plasmogamy, then second is the karyogamy, third one is the meiosis. So karyogamy and meiosis takes place in the basidium, okay, leading to the formation of 4 basidiospores. Okay, this we have to remember, 4 basidiospores are formed, whereas in ascomycetes 8 ascospores are formed. Okay, 8 in ascomycetes and 4 in basidiomycetes, and these spores are produced in basidiomycetes, whereas they are formed in ascus in ascomycetes. Now the basidiospores are exogenously produced on the basidium. Okay, now the basidia or the basidium are arranged in the fruiting bodies called the basidiocarps. So the fruiting body in basidiomycetes is known as basidiocarp, whereas fruiting body in ascomycetes is known as ascocarp. Okay, ascocarp is the fruiting body in ascomycetes, and these fruiting bodies are born on the basidia here in basidiomycetes, or basidium you can say. Okay, now this is the life cycle of a member of a basidiomycetes. Again, this is very simple. Here you can see germinating basidiospore. Basidiospores are formed here. Let's start with here, homokaryotic mycelium. Okay, means which contains one nucleus, which is haploid. Okay, homokaryotic mycelium. Plasmogamy takes place here, means the fusion of the protoplasm. Then what happens? Dikaryon is formed, which is also known as heterokaryon. Okay, n plus n. This is a dikaryon here, which is also known as heterokaryotic phase. Okay, and then what happens? Karyogamy, fusion of nuclei will take place. These two nuclei will fuse together. Okay, in the basidiocarp. Here, this is the basidiocarp, fruiting body. Okay, and then what happens? A diploid stage is achieved. Okay, so basidium is diploid, and then meiosis will take place here, leading to the formation of basidiospores, and these basidiospores will germinate to form the hypha. Okay, here you can see the basidiospores are produced exogenously. These are basidiospores, they are forming exogenously. These are basidiospores, okay, which are produced exogenously in the basidium. Okay, so meiosis takes place in the basidium. The example of basidiomycetes, first one is the agaricus. Common name is mushroom. This is agaricus. And then we have ustilago, which is also known as smut. Okay, then we have Puccinia, which is known as the rust fungus. Okay, so these two are harmful one, ustilago and Puccinia. This is ustilago, this is Puccinia. Here you can see it is causing some spots on the leaf of the plant. Okay, these are the spots. Now let's talk about the Deuteromycetes. So Deuteromycetes, one thing you have to remember, they are known as fungi imperfecta. Okay, commonly known as imperfect fungi. They are known as imperfect fungi. Why? Because the sexual stage is missing. Is the sexual stage has not been found in Deuteromycetes. That's why they are known as fungi imperfecta or imperfect fungi. Okay, so here I have written because only the asexual or the vegetative phases of these fungi are known. Okay, and sexual stage is absent here. Okay, but what happens as we go on studying the organism? Okay, what happens, sometimes we find the sexual stage. Okay, all right, earlier the sexual stage was not formed, but as we are developing new techniques and all. Okay, so sometimes we find out the sexual stage, and whenever sexual stage has been found out, then what happens? Those members will be shifted to either Ascomycetes or Basidiomycetes. Okay, so once perfect or the sexual stage of the member of the Deuteromycetes is discovered, then what happens? They were often moved to Ascomycetes or Basidiomycetes. Now, general characteristic of Deuteromycetes: mycelium is septate or branched. Nutrition can be saprophytic or parasitic. However, large number of them are decomposers, okay, of litter and help in mineral cycling. So this is the importance of members of Deuteromycetes. Reproduction: the Deuteromycetes reproduce only by asexual spores called conidia, and the sexual stage or the sexual reproduction is absent. Example of Deuteromycetes, this one is very important: Alternaria. Okay, then we have Coritotrichum and Trichoderma. Trichoderma is also very important. So most of the Trichoderma is beneficial. Trichoderma is known as beneficial fungus or biostimulant. Okay, this is an image showing the Alternaria. So these are the spores of the Alternaria. These are the asexual spores because sexual spores are absent. This is Colitotrichum. You can see spots here on the leaves. Then we have Trichoderma. This is the microscopic image of the Trichoderma fungus. Now let's talk about the kingdom Plantae. The general characteristics, as we all know, kingdom Plantae include eukaryotic organisms and they contain chlorophyll, okay, which helps in the photosynthesis. Now, a few members of kingdom Plantae are partially heterotrophic. Okay, usually the plants are photoautotrophs. Okay, they can prepare their own food, but there are some exceptions. Okay, some of the plants are heterotrophic. Okay, example is insectivorous plant or parasites. Okay, now bladderwort and Venus flytrap. Okay, these are the example of insectivorous plants. Okay, they feed on insects. Okay, and they attain nitrogen from them. All right, and the example of parasitic plant is Cuscuta. This also you have to remember, question can come from this. Okay, then the question they will ask you example of insectivorous plant or the example of a parasitic plant. Now the plant cell have a eukaryotic structure. They are eukaryotes with prominent chloroplast and cell wall is mainly cellulosic. This also you have to remember. Plants have cell wall which is made up of cellulose. Now classification of kingdom Plantae. The kingdom Plantae are further divided into algae, bryophyte, pteridophytes, gymnosperm and angiosperm. So you will study about all of these different categories or groups in the chapter kingdom Plantae, which will be the next one. Now classification of kingdom Plantae. Now the life cycle of plants have two distinct phases. This you have to remember. The life cycle of plants have two distinct phases. First one is the sporophytic phase, okay, which is diploid. Okay, consists of two sets of chromosome, and the other one is gametophytic phase, which is haploid N. Okay, and these two phases alternate with each other. Okay, so plants show alternation of generation. Okay, first phase is the sporophytic, second is the gametophytic, which is haploid, and these two stages alternate with each other. Okay, one can be dominant, other can be dependent on the other phase. Okay, Now, alternation of generation. So what happens? The length of the haploid and the diploid phases, means the gametophytic and the sporophytic phase in the life cycle of the plants, okay, and whether these phases are free living or dependent on others, vary among different groups in plants. Okay, so here they are saying that the length of the gametophytic and the sporophytic phase, okay, it varies. Length of the phases varies, as well as whether these phases are free living or they are dependent on others. Okay, this also varies. Now let's talk about kingdom Animalia. We will study about the basic features of kingdom Animalia, not much. So the basis of classification of animal of kingdom Animalia, okay, first one is that they are heterotrophic eukaryotic organism. Okay, the plants are usually phototrophic, animals are heterotrophic organism that are multicellular and they lack cell wall. Okay, plants have cell wall, but the animal cells lack cell walls. What are general characteristics? They directly or indirectly depend on plants for food. Okay, directly or plant. Okay, herbivores. If we talk about herbivores, they are directly dependent on plants. But if we talk about carnivores, they are indirectly dependent on plants, because carnivores feed on the herbivores, okay, and herbivores feed on plants. Okay, so carnivores are indirectly dependent on plants. And then what happens in animals? They digest their food in an internal cavity. So they have the internal cavity. They digest food in the internal cavity. First of all, they ingest food, okay, and then what happens? They store the food reserves as glycogen. So this also you have to remember, the reserve food material in animals is glycogen or sometimes fat, but usually it is glycogen. The mode of nutrition, this you have to remember, it's holozoic in animals, means ingestion of food. We ingest food, okay. So the higher forms show elaborate sensory and neuromotor mechanism. So higher forms like humans, they have elaborate sensory and neuromotor mechanism, whereas the lower forms, okay, like hydra and cockroaches, if we talk about the insects, okay, then they, in that, the sensory and the neuromotor or the nervous system is not that much developed. Okay, now most of them are capable of locomotion, they can move. If we talk about the reproduction, how does sexual reproduction occurs in the higher forms? Okay, so sexual reproduction is by the copulation, or you can say the fusion of the male and the female gamete. Okay, male and the female gamete, they fuse together, leading to the formation of zygote, and zygote forms an embryo. Okay, so that's why embryological development, this is also a feature of kingdom Animalia, or the members of kingdom Animalia, embryological development. Now let's talk about, this was the end of the 5 kingdom classification. All right, now we will discuss about the viruses, viroids, prions and lichens. Okay, so let me give you a brief introduction. Okay, so in the 5 kingdom classification of Whitaker, okay, there was no mention of lichens and some acellular organism. Okay, so Whitaker didn't mention lichens, and also he didn't mention the acellular organisms. Okay, organism which doesn't contain cells, which are acellular, like viruses, viroids and prions. Okay, so Whitaker, okay, didn't included these organisms in his system of classification. So viruses, let's talk about viruses. So viruses, they are considered not considered as truly living, okay, because first they are acellular. All right, so viruses did not find a place in the system of classification, okay, since they are not considered truly living. So they are, you can say viruses are obligate parasite. They need host to complete their life cycle. They cannot survive without a host. And whenever they are inside the host, then they are living. Okay. Inside the host, they are known as living. Okay. But outside host, they are non-living. Okay, so here viruses did not find a place in classifications because they are not considered to be living. If we understand that living as those organisms that have a cell structure, so they are acellular. Now, the viruses are non-cellular or acellular organisms that are characterized by having an inert crystalline structure. So, they have an inert crystalline structure outside the living cell, means outside the host. Outside the host, they survive as inert crystalline structure only, which is non-living. So, they are the connecting link between living and non-living viruses. Now, let's study about the general characteristic of viruses. Okay, so once the virus infect the host cell, they take over the machinery of the host cell to replicate themselves, and then they kill the host. Okay, now discovery of virus. Let's study about the discovery of the virus. Now the name virus means venom or poisonous fluid. Okay, so there was one scientist, Dmitry Ivanovsky. So what he did in 1892, he was working on the tobacco plant. So what he recognized or observed that certain microbes, okay, are the causal organism of the mosaic disease of tobacco. So what he did? So this is an image showing the tobacco mosaic virus. All right. So this is an RNA virus. The DNA material or the genetic material is RNA. Okay, and this is the capsid. I will explain you what is capsid in upcoming slides. Okay, so now for now you have to remember tobacco mosaic virus is a plant virus and it consists of RNA as a genetic material. So what he did, Ivanovsky? So he observed that, or he found out that these entities, okay, these entities mean these biological entities, okay, or the pathogen or whatever, these are even smaller than bacteria. So now we know viruses are even smaller than bacteria. So these entities were smaller than bacteria because they passed through the bacteria proof filters. So what he did, he passed that filtrate which he obtained from the tobacco plant, okay, through a bacteria proof filters, okay, which can filter the bacteria, okay, but it was not able to filter these biological entities. All right, so he said that these entities are even smaller than bacteria because they can pass through the bacteria proof filters, and if applied on the tobacco plant it can cause disease. Now next scientist, okay, his name was Bejering. So what he did, Bejering, he demonstrated that the extract of the infected plants of tobacco, so he was also working on tobacco. So what he did, he took out the extract from the infected plants of tobacco and he said that it can cause disease in the healthy plants as well. Okay, and he named that extract as, okay, contagium vivum fluidum. So this is very important. Okay, he named the new pathogen virus. Okay, he named that extract or the pathogen as virus and called the fluid. Fluid means that extract. He named that extract as contagium vivum fluidum and he named the new pathogen as virus. Now, later what Stanley did, W.M. Stanley, he showed that the viruses could be crystallized. So he was the first person to crystallize the viruses, okay, or you can say he showed the crystalline structure of the viruses, okay, and the crystal of virus or the inert crystalline structure. We are talking about the structure of the virus. It consists largely of proteins, okay. So the viruses are made up of largely of proteins, okay. So one part is protein, another part is the genetic material which can be DNA or RNA, okay, RNA or DNA, okay. So that's why we call viruses as nucleoprotein. Nucleic acid, RNA and DNA, nucleic acid, this is protein. So viruses such as bacteriophage are known as nucleoprotein. They consist of nucleoprotein. So they are inert outside the specific host. This we already know. Now the viruses are obligate parasite. This also I've already explained. Now in addition to proteins, viruses also contain genetic material which can be RNA or DNA. That's why they are known as nucleoprotein. Okay. Now no virus contain both RNA and DNA. They will either contain DNA or RNA. So the viruses is the nucleoprotein and the genetic material is infectious. The genetic material of viruses are infectious. Now in general, viruses that infect plants, this you have to remember, have single-stranded RNA. Okay, and viruses that infect animals have either single or double-stranded RNA or double-stranded DNA. So DNA is double-stranded, whereas RNA can be single or double-stranded. So the bacterial viruses or the bacteriophage. So what are bacteriophage? Which infects bacteria. Okay, bacteriophages are viruses which infects bacteria. Okay, viruses that infect bacteria are usually double-stranded DNA viruses. This also you have to remember. Now let's talk about the structure of virus. The protein code called the capsid. I've already showed you in the figure what is capsid. So the capsid is known as the protein code. It is made up of small subunit called capsomers, which protects the nucleic acid. Okay, I'll draw a rough diagram of a bacteriophage. Okay, so here the nucleic acid is present. Okay, this is the entire structure of the bacteriophage. Nucleic acid is present here, and this is known as the capsid. Okay, which is also known as the protein code. It is made up of proteins and it is made up of small subunits. Okay, so these small subunits are known as capsomers. So these capsomers are arranged in helical or polyhedral geometric forms. So this is an image showing you a bacteriophage. So here this is known as the head, capsid head. This is the nucleic acid which can be RNA or DNA. This is the collar here. Okay, these are the whiskers. This is the sheath. Okay, this is the base plate here. This is the base plate. Okay, these are the tail fibers and this is the spike. Okay, and this is the 3D image of a bacteriophage. Again, this is the capsid. Nuclear material is present inside here. Okay, this is the collar here. These are the tail fibers and all. Okay, so this capsid, okay, also known as the protein coat. So this is made up of protein coat. Okay, and it is made up of subunits. What are the subunits called? Capsomer. Now diseases caused by virus. Okay, viruses cause diseases like mumps, smallpox, herpes and influenza. So influenza is very common. Okay, it is caused by the viral infection. Herpes also is caused by virus, smallpox. Okay, in humans, AIDS is also caused by virus. And what is the name of the virus? HIV, human immunodeficiency virus. Now disease again, in plants. Okay, the symptom of viral infection can be mosaic formation, like in tobacco mosaic virus, leaf rolling. Okay, plant viruses called leaf rolling and curling. Yellowing of the leaves means there is a degradation of the chlorophyll, and vein clearing, dwarfing and stunted growth. It can affect the growth also. Now let's talk about the viroids. So viroids were discovered by T.O. Diner. Okay, so he discovered a new infectious agent that was smaller than viruses. Okay, and caused potato spindle tuber disease. Viroids were found to be free RNA. This you have to remember, that lack the protein code, hence name viroid. The RNA of viroid was of low molecular weight. This also you have to remember. Now let's talk about the prions. So prions are what? They are infectious protein. Okay, infectious. You have to just remember prions are infectious protein particles. Okay, and they can cause lot of neurological diseases. Okay, and what does prion contain? They are just the proteins. Now the agent was similar in size to viruses. Okay, so they are similar to sizes of viruses. The most notable diseases caused by prions are bovine spongiform encephalopathy. They cause encephalopathy. What does encephalopathy means? Okay, it is a neurological disorder. Your brain is not able to work properly. Okay, commonly called as mad cow disease. So BSC is also known as mad cow disease because, okay, so it is seen in cows, okay, and cattle, and its analogous variant. Okay, analogous variants mean functional similarity is CR Jacob disease, which is seen in humans. It is also a neurological disease. Now, these are the differences between viruses, viroids, and prions. Okay, so viruses, you can see this is definition of viruses. Viroids are RNA particles. Okay, these are the prions. They are simpler infectious particles made up of proteins. This you have to remember. They cause a lot of infections. Okay, special about prions, they cause neurodegenerative diseases. Okay, this you have to remember. Examples also you have to remember. Now lichens. Lichens I've already told you, it is symbiotic association between algae and the fungi. Okay, it consists of algal component and fungal component. Algal component is known as phycobiont. Fungal component is known as mycobiont. Now algae in association with fungi prepare food. Okay, so algae provide food to the fungi and fungi provide shelter to the algae. Now, what is the importance of lichens? Lichens are very good pollution indicator. This you have to remember. Okay, and they do not grow in polluted areas. Okay, lichens can only grow in pollution-free areas. That's why they are good pollution indicators. Now, let's talk about the previous year questions. Okay, so I have given you these questions. Okay, there are 10 questions. 5 we will solving here, and rest of the 5 will be your homework exercises. Okay, so these questions I've tried to include different type of questions here. Okay, some are direct and some will be statement questions. Okay, so first let's start with the first question, which is the imperfect fungi that are decomposers of litter and help in mineral cycling. This I've already told you in the slides. Okay, options are Basidiomycetes, Deuteromycetes, Ascomycetes, and Phycomycetes. So simple, this is a very direct question. Answer is option B, Deuteromycetes, which are also known as fungi imperfecta. So these, the members of these fungi, okay, helps in mineral cycling. They are decomposers. The next question is, here we have to pick the incorrect statement. So you have to see the question correctly. Sometimes they will ask you to pick the correct statement and sometimes incorrect statement. Okay. Now Neurospora. So Neurospora, which is the haploid fungi, is used in the study of biochemical genetics. And okay, so this should be true. Penicillium is multicellular and produce antibiotics. We have already seen penicillium produce antibiotics. Alright, and it is multicellular. Yeast is unicellular, used in fermentation. This also I have told you. This is also true. Okay, this is used to make bread and okay, so, and it uses the fermentation to produce this bread and all. Alright, next option is morels and truffles are poisonous mushroom. Okay, you have to read every statement correctly. Here they are saying they are poisonous. So what should be the incorrect statement? Morels and truffles are poisonous mushrooms. Okay, so this is incorrect statement because actually they are edible. They are edible mushrooms. They are not poisonous. Now let's come to the next question. Third one. Here you have to identify the fungi which do not belong to the group of other fungi. Okay, options are sac fungi, puffballs, mushrooms, and bracket fungi. Okay, so this I've already told you. Let's talk about Basidiomycetes. So Basidiomycetes, what are they commonly known as? Okay, or what does they include? They include puffballs, they include mushrooms, and they include bracket fungi. Okay, so we are left with the option A, sac fungi. So this is the odd one out. Okay, so this doesn't belong to the other group. Okay, so all these are the member of Basidiomycetes. And what should be the answer? Sac fungi. Sac fungi are Ascomycetes. Okay, these are Ascomycetes, whereas puffballs, mushrooms, and bracket fungi are All these three are basidiomycetes. Okay, now let's talk about the mad cow disease. Okay, mad cow disease in cattle and the CR Jacob disease in humans are due to infection by, okay, options are bacterium, virus, viroid, and prion. Okay, so bacterium, it is not caused by bacteria, we've already studied. Okay, then it is not caused by viruses also. Okay, so we can remove these options. Viroids and prions, it is also not caused by viroids. So mad cow disease is a neurodegenerative disorder. Okay, it causes encephalopathy in cattle and it is caused by prions. Okay, so prions are what? They are infectious agent. Okay, and they are made up of proteins which are similar in size to viruses. Okay, now coming to the next question. Here they are asking, this is again a statement question, and here they are asking about the correct statement. Which of the following is correct about the viroids? Okay, options are they are free RNA without protein coat, they have DNA with protein coat, they have free DNA without protein coat, or they have RNA with protein coat. So we have already studied about viroids. Viroids are, they contain the genetic material. Which one? RNA or DNA? Okay, you have to see. Okay, you have to remember either they contain RNA or DNA. Without protein coat, that we know. Okay, they do not contain the protein coat. It's a free living genetic material. So what should be the answer? They have free RNA without protein coat. They don't contain a protein coat. Protein coat is an characteristic feature of what? Bacteriophage. Okay. So the viroids, they have free RNA, okay, without the protein coat. Now, these are the previous year questions I have listed here, but this is your homework exercise. All right, you can have a look at them. So this question is coming NEET 2019. Okay, and you can go back and read these questions and answer. Okay, you can watch the video again and you can comment in the section. Second one is this: which of the following statement is correct? Again, these are your homework exercise. Viroids differ from viruses in having, okay, this we have already discussed. Viroids differ from viruses in having what? Protein coat. They don't have protein coat. Then which one of the following is wrong for fungi? Okay, this has come in NEET 2016. Okay, these are your options. Please do comment if you are solving these questions. We will revert back. Now, which next is which one of the following statement is wrong? This is again a statement question. All right. So this was the end of this lecture. Okay. We will start with the another chapter tomorrow. Tomorrow will be day 4 of the NEET Biology lecture series. And hope you enjoyed this session. We will meet again. Thank you very much for listening.